Did you know that it took over 13 years and a $3 billion investment to sequence the first human genome? And now, with all the latest tools and infrastructure available, we can achieve that within an hour. But there are still customers out there, they are spending over weeks and months to perform the genome sequencing on human genome and on different type of data sets because they are stick to the traditional workflows that utilizes the CPUs and they take care of all the other things that are slowing down the process. The first challenge is genome sequencing and the output coming out of these are in terabytes, huge terabytes. Like uh, starting from just the input size, you can assume it to be 2 to 3 terabytes, and the output would be in GBs. And plus, to make this compute and storage to operate better, you need a good networking solution. So, in the current market, it's all about time to market, and like if we need to start fast, fail fast, so that we can restart. The high level challenges faced by our customers today. The more crucial was the high energy utilization. So imagine you are running something at scale utilizing hundreds of CPUs and different servers. So that is consuming a lot of energy. And also the failure recovery and failure handling is very difficult. And the tools that they are currently using, they are not compatible to run on GPU. So they cannot be accelerated directly on GPU. To give you a brief overview about the genome sequencing, you can see the two parts. On the left-hand side, this workflow is something that is done manually, still done in the lab by the technician. This cannot be automated. So the flow on the left-hand side is called the primary analysis, which is carried out in the lab. On the right hand side, that's the computational analysis that is called the secondary analysis. And our focus is accelerating this particular part because we have some data that can be accelerated using GPUs and infrastructure. And the next slide is explaining you the high level flow, what's exactly happening. So you get a sample, you isolate the DNA from that particular sample, you fragment that particular sample for the preparation for the genome sequencing, you enrich that particular sample using some specific techniques, and then you amplify it so that your sample is very clear. Then it goes into a specific machine, and that particular machine translates uh, that particular sequence into something that can be understandable by the computers. You can think of it as a huge text containing A, T, C, and G. So these all are nucleotides, and I'm not going to go into much deeper, but you can assume it to be a text that can fill over 200 New York City phone book. So that text you need to compare with another reference genome. So you can think of it as a huge string comparison, where the first step is to map and combine all of those DNAs together. That part is called the read alignment. And that's why you see here the sequencing machine on the left-hand side, you see the raw data that goes in, that is transformed into the FASTQ files. Then you run into the BAM file, and then you run uh, basically you do a alignment of the data, and once the data is aligned to the original DNA, then you do a comparison, like to identify the variant, what is the differentiation between them. So that part is called the variant calling format. There are two parts that the bioinformatician work on. Their pipelines are the germline pipeline to identify the issues that is heredity, like that is coming from your genetics. And the second one is one that you build during your lifetime. Bioinformaticians, they were very comfortable running in an environment where they wanted a terminal access, where they can simply execute their workflows and scripts, and they're not very familiar with Kubernetes layer. So we have created a custom image that can run on private cloud AI, and you can launch a notebook using that custom image, and that particular notebook will have all the dependencies resolved, and it will have inbuilt NVIDIA Parabricks. The good part here is it can be run on GPUs. They can be GPU accelerated. So the solution here is notebook that is containing NVIDIA Parabricks, and on top of that, it has all the necessary tools required by the bioinformaticians to run their traditional pipeline plus the GPU accelerated pipeline. So that image itself is the complete solution. This is just a foundation for the customers to get started to enable them from day one. Before going on to the target outcome, I will share the steps that you need to follow to launch the notebook. I have created a custom image which is built on top of PyTorch CUDA base image, and that particular image is combining the CPU-based tools plus NVIDIA Parabricks and some necessary libraries that is required to do the end-to-end bioinformatician genome sequencing pipeline. So for that, what you do is you click your custom image, you give the image path, you go to the UI, you go to either the Kubeflow or the notebook section, click on notebook server, define the name, select you want to run the Jupyter Lab, enter the custom path. So the image that you see here, this image is built on top of AI Essentials 1.6. It's an older version, but I have tested it. It still works in the latest version of AI Essentials, which is 1.10. So it's not recommended to use it for the production purposes. It's recommended that you rebuild this particular image on top of the latest AI Essentials image, but it still works and you can use it for the demo purpose. So image can be hosted either locally, for example, in private cloud AI, you can deploy your own image registry and give that particular path. Or you can upload it to your Docker Hub and you can pull it from the Docker path, Docker Hub, given the path for the Docker Hub here. And then what is the resource required? What is recommended for NVIDIA Parabricks to operate is you use minimum of 64 CPUs, minimum 64 GB of RAM, and at least 2 GPUs. And when creating the notebook, here you select what's your workspace volume that should be mounted in your notebook. So you can define 150 GB. I have used it for sample, but you can choose to have a little bit more in case you're pulling the data from internet that will require at least 200 to 300 GB of data. So I recommend putting it at 500 GB and read-write many for better performance. And at the end, you can also, when creating the notebook, you can also mount an existing volume. For example, one of your peer has created a common volume where all this data is located and you want to share it with other person. Either you can use the shared volume and then you can launch genome sequencing. So here you see CPU workflow script, data preparation script, GPU workflow script, prepared environment. So when we start a notebook, we first need to run this particular script. This script will set the environment, like setting up your LD library path, setting up path, and making sure when you create multiple terminals, then your environmental variables are saved in your bash profile so that it is persisted across multiple terminals. So I'll show you a quick glimpse of what is there in the prepare env. This can also be embedded within the container image. Resetting the environmental path to make sure that all the necessary libraries are set in the LD library path. And when you run any of the executables, it should be able to run without specify the full and absolute path. The second part is the data preparation. So once your environment is set, here in this case, let's just take this one as an example. So this is like A. theliana, a plant-based genome, which is majorly used for the demo purposes. So you have a data preparation script. You just need to run this script and it will pull the necessary data. And among the data, the most important thing is these two files. So these are the reads that is generated by the sequencing machine. So these all are nucleotides and it comes in a specific pattern. For example, A and C will always come together, G and T will always come together. It's like two type of nucleotides. They will be always combined together. This is your reference genome, the FASTA file. So the first step is read alignment, where you give this as input and give the reference genome. I'm going through the CPU workload script, and I have identified in such a way that in each one you have a different folder for different type of genome. I'll also show how it looks like in with the GPU accelerated using NVIDIA Parabricks, but first I'm showing you how it looks like with the standard CPU-based tool. So you give your FASTA file, which is your reference genome, and then your two FASTQ files. You specify the number of threads it should use, then you run the FQ2. It depends on the resources that you have available. You can tune these numbers. And then you do BWMM, you do a mark duplicates, then you run the indexing tool, and generate the final BEM file. You can think of it as a regenerated full genome from the samples which was fragmented earlier. Now I'll show you the sample execution looks like. So the total execution time was 3 minutes, 54 seconds approximately. And if you scroll up here, the execution is huge. But here you will get an idea what samples it was currently processing, what state it was running BWMM at this particular point. And then once that was done, how much time the BWMM took. Oh boy. Then it went to the marking duplicates and generating the index file. And towards the end, you will get the total execution time. Now let me go back to the GPU execution of the same script. So in the GPU execution using NVIDIA Parabricks, there's a PB run command, which is Parabricks run, and you run fq2bem. And previously you were running 3 different steps: read alignment, marking duplicates, indexing. Here everything is done through single line of code. You don't need to run a function. You don't need to specify multiple threads. You just need to specify number of GPUs that you want to use. So once you execute it, it will generate this file. And here you see the total execution was 1 minute 34 seconds. And this is run through the pbrun haplotype caller. You give the reference genome, what output it should generate, and the output variant, and the number of GPUs. And here to use low memory option, I mean, you can use HTVC low memory. And of course, when you run pb run -help. So this is what claimed by NVIDIA. And of course, this results will vary based on the type of hardware you're using and the infrastructure you're using. So you can see that 18-hour plus that can be minimized to 1 or 2 hours. So what I have worked on 3 different data sets. The first 2 data sets are the downscale version that are good and sufficient in case if you want to demo it to any of the customer. So there we have seen an acceleration up to 18x to 19x. But then we tried running something similar, which is not the downscale version. So we got this request that can we run this acceleration on the full human genome. So they suggested HG19, and we ran not just the 2 use cases, but some additional use cases that might be useful. And there we could see the acceleration up to 27 times. And of course, the customer was was very happy seeing these results, and they wanted to run this now on their private data set.